I'm Peter McKay. I'm Curator of Contemporary Australian Art here at Cragoma, and I've also been the curator ushering this immense project into the gallery. I'd also like to begin by acknowledging the traditional custodians of the land upon which we stand and pay my respect to elders past, present, and emerging. Thank you for joining us here today at Patricia Piccinini Curious Affections opening weekend. This conversation is presented in association with World Science Festival. It does have a bit more of a science theme than some of the other programs, but that's probably why you're all here. Today, I'm joined by the artist, Patricia Piccinini. I'm sure you all recognize her by now. We also have guest speakers, Eben Kirksey and Elizabeth, or Ella Finkel. Please. Great. Elizabeth is a former biochemist, and she is editor-in-chief of Cosmos magazine and publication contributor to our exhibition catalogue, which I might add, I'll plug it a couple of times throughout this talk, but I might add it will arrive in about a month's time, before the end of April, we hope, and is available for the very reasonable price of $44.95. Eben is Associate Professor, no less, at Alfred Deakin Institute at Deakin University, and is best known for his work in multi-species ethnography. So we'll all understand what that is by the end of this talk. In this conversation, we'll be discussing the intersection of art and science around this exhibition. So please help me once again, I know you already have, but welcome our speakers to the stage. So Patricia, I'd like to start with you. I know you've been talking a lot this weekend already, but I thought it would be great if you could talk a little bit about the presence of science in your work and your practice, because obviously that's a great thematic departure point for us now. How would you like to do that?
Okay. I see science as the way that we tend to predominantly understand the world around us. It used to be mythologies, all kinds of mythologies, pagan mythologies, Greek mythologies, then it became religion. And now, even in my lifetime, I'm 50, it's become, I don't see the world in terms of religion, I see it in terms of science. And I think that's quite a good thing. But it is just a way of seeing the world. And I use these science stories to talk about sort of broader, more ethical concepts that I'm more interested in, or I think are more vital. I just see science as a given. As an artist, I'm not trying to say that science is good or bad. In fact, what I'm trying to do, I'm trying to have a conversation through the artwork with my community about the implications of science and how science changes the way we see each other and our relationships, the relationships between what we consider natural and what we consider artificial, the relationships between us and other animals, between us and the environment, us and our bodies. I think science changes those relationships dramatically. And in my work, I don't present myself as someone who knows a lot, cognoscenti, someone who knows a lot about science. I'm not. I really am not. But I'm interested, and I read a lot about it, and I'm just as confounded as everybody else, and I'm just trying to make sense of these dramatic things that happen around us.
I think that's a really interesting point, actually, because we often think that culture shapes our worldview. You know, there's always so much discussion about, I don't know, violent movies or video games and things like that. But actually, the idea that science is a really omnipresent guiding force of the way that we make sense of our world, I think it's maybe so evident that maybe we forget about it sometimes. But continuing on from that a little further, are there some specific technologies that have been important to your practice that we might introduce into the mix?
Oh, sure. Look, I think we're living through the most amazing time. I did a work in 2002 about xenotransplantation, which is growing human organs in other bodies, in other animals. And that's when I made the young family that's in that room there. And I mean, along the way, I've made work about protein, about tissue engineering, stem cell research. I mean, there's just so many. At the moment, it's CRISPR. CRISPR's a big deal.
Can I ask, how many, this is an interesting crowd, I think it's an arts crowd and a science crowd. How many of you know about CRISPR? Hey, hello, that's amazing. But sorry, Patricia, please continue. I just wanted to gauge that.
Oh, okay. Yeah. Well, CRISPR is a gene editing technology. It was developed about 4 or 5 years ago, and it means in California, it just means that gene editing is become incredibly not easy, but it's easier to do and very inexpensive. And so it's allowed a lot of people to do fast track a lot of research. And they say that this technology will be part of every birth in the next decade. That's a big thing. Yeah. So, I mean, I'm no expert. But I'm vitally interested in what it means as a community to be able to change and choose how our progeny will come into the world and what they will be like, and what that means for a whole bunch of other things, like how we relate to difference. Will we tolerate difference? Will we become a much more homogenized society? I'm interested in those things.
That's great. Well, maybe, Ella, I think you are the brains trust here in terms of explaining, understanding some of this history of genetic modification and stem cell research and CRISPR, the histories of these things and how we've come to terms with them. And in fact, that's why I really wanted you to write for the catalogue, because the context in which we understand Patricia's work, I think, has changed quite a lot. So those first works that you were doing, like psychotourism, which primarily about stem cell research, if you think back to the mid-90s when that work was made, there was a lot of discomfort, there was a lot of hysteria, there was a lot of panic and a lot of misunderstanding about what this technology might promise. And the technology has changed, we seem to be more comfortable with it, but Ella, you're not always so sure about that, are you? That we're, just as we get something comfortable with something, it becomes a bit more, there's a whole new frontier that opens up to worry about. But that's a very long-winded way of asking, could you talk to us a little bit about stem cell research and CRISPR and these technologies that Patricia uses as a premise?
Okay, thank you, Peter. So I was very honoured to be given the invitation to write an essay about Patricia's work. And while at the beginning I thought, oh, what do I know about art criticism? Can I learn this really quickly? I soon realised Peter didn't expect me to be the art critic here, and asked me to reflect on the work as it related to a book I'd written around the same time, Stem Cells, Controversy at the Frontiers of Science, that was published just a little bit after Patricia had her exhibition at the Venice Biennale, the first Australian to exhibit there. And so this was a great opportunity for me, and it's resulted in quite a significant journey for me, contemplating art and science and ethics and the whole shebang. But maybe I can just quickly reprise that journey for you just in a few minutes. So I see that Patricia and I were both responding to a revolution that took place shortly after 1998. So today it's 2018 and we're celebrating, or not, the 20-year anniversary since scientists first learnt to make human embryonic stem cells. It was an incredible discovery. It gave us incredible abilities, and Patricia could see that, and she responded to it artistically in the works you see in her gallery. And I responded to it scientifically. I ended up writing a book about it. And a lot of my friends knew that I was writing this book and said, oh, you should check out Patricia's work, you know, on still life with stem cells and the young family. So I did at the time. And I have to say my reaction was not positive at the time because I felt that it was dystopian, that she was, and this was exactly what I was trying to combat in my book. But it's been a journey for me. So let me get back to my train of thought. So let me tell you a little bit about what scientists did in 1998, 20 years ago. So they had taken embryos from IVF clinics, embryos that were surplus and would normally be thrown away. And they had learnt to take from those embryos those very specialist cells that can give rise to any of the organs of a human being. Those cells only exist for a very, very short time, maybe for a few days, and then they turn into the various body parts. But scientists had learned to freeze them in that primordial state so that they were basically stem cells on tap. They would grow forever, and you could then train them to become whatever body part you wanted them to. And I like to think of it in terms of a car. So if you have a car, you know, your brakes fail, you replace them. The tyre, you know, burst, you replace the tyre, so on and so forth. And if you keep doing that, you can pretty much run that car forever. And stem cells meant we could start thinking of human beings in the same way. You need spare parts. You know, perhaps your child has diabetes. We can grow that child a new pancreas. Perhaps a person has been in a car accident and their spinal cord is damaged. We can grow the cells to repair the spinal cord. Perhaps someone a little more elderly has Parkinson's disease or heart disease. We can replace those cells, keep that person pretty much running, you know, in pretty good condition for a long time. I found this, as I say, Patricia responded artistically in that holistic way that artists do. Artists are charged with fulfilling that impulse to examine the human condition, and the human condition had obviously just changed profoundly. And I, as a scientist, wanted to really convey to the public how extraordinary this discovery was. And one of the things I realised I had to do in my book was to talk about the ethics. And I guess I was quite surprised by the strength of the backlash against the stem cell revolution. And this ethical backlash was fueled by two things. One, by a sort of a religious conservatism, very much driven by people of the Catholic faith who view an embryo as a ensouled person. And the other came from the slippery slope type argument, that if you were to allow this kind of research to continue, then where will it end? Will we have designer babies, as Patricia imagined? Will we be able to mix these human stem cells with animal stem cells and make the pig-human mother that Patricia imagined in her young family, and so on? And in my book, I felt, you know, I could address that concern very easily by saying, no, the slippery slope doesn't exist. We have checks and balances. And my main argument was to say that actually we've had the tools to do designer babies for a long time. We've had the tools to do cloning for a long time. because, you know, we clone animals and we can do genetic engineering in animals, but it hasn't happened in humans. And I argued, well, that's because we have checks and balances. So rest assured, we can go ahead with the stem cell revolution and we don't need to worry. And in fact, that's pretty much what happened. I think ethical bodies all around the world, regulatory bodies in Australia particularly, we passed legislation, very strict legislation with checks and balances. So it's 20 years on. What's happened? I can tell you that last week, we had the first really big clinical entry of something from stem cells. And that was that 2 people who would have gone blind regained vision because of a stem cell graft into their retina. You know, for me, the most fascinating thing is that it took 20 years. Because when I was writing my book, one of the questions I would ask the scientists, or what everybody wanted to know, well, okay, you've got this raw material to make new body parts. How long is it going to take till my child can get a new pancreas, till we can fix my son's spinal cord, till we can fix my husband's Parkinson's? And the scientists were sort of, well, you know, 5 years, hard to say, maybe 10, 20 years. But when you think about it, all construction projects run over schedule, don't they? And we have a pretty extraordinary construction project here. So it's taken a long time. The only cab off the rank so far is fixing this particular form of late age blindness, age-related macular degeneration, which 1/3 of people as they age will experience. The other cabs are still in the rank, still in the pipeline, getting replacement cells for diabetes, for Parkinson's disease, and so on. People haven't given up, but it takes a long time. So we're getting the results much slower than anybody thought, I think. And what about the checks and balances? What about the slippery slope? So I have to eat my words here and say, okay, Patricia, you were right. Because the things that I argued in my book wouldn't happen are happening. Because it turns out the reason why nobody was doing genetic engineering of humans was that the technology we had, genetic engineering, was just too crude a tool, too crude to apply to the precious embryos that we can get from IVF clinics. Okay to do in plants, okay to do in animals where you can do 1,000 goes, but not okay to do on humans where the embryos are very precious. But now we have this new tool, CRISPR, so precise, we don't call it genetic engineering anymore. It is so fine and specific, we call it genetic editing, and suddenly everything's changed. In 2015, Chinese scientists started trying to genetically engineer human embryos, still in the lab, but the research has been going ahead apace. And then last year, I was quite taken aback to read that people had started making human-pig chimeras, embryos anyway, still in the lab, but for a good reason. Turns out, 20 years on, we still haven't managed to make anybody a pancreas. It's a little harder than we thought. And if you could put human stem cells inside a pig, then it would be easier to make replacement organisms. It's easier to do it that way. So good reasons. But I started off this little dialogue by saying I've been on a journey since I've been invited to write this essay, and I realised that technology changes and it changes the scientific landscape, and you know what? It changes the ethical landscape. And knee-jerk answers don't work. We need to look at these new technologies, look at it and see what is possible. And artists like Patricia, who have been doing this for 20 years, examining the human condition, examining how people respond to things, how they respond to orifices with hair on them, and all the other things that are part of our deep psychology, I think, are very valuable.
I'd say this is a highlight too.
But you know, in response, I don't feel that a work like The Young Family is a cautionary tale. I don't feel like I am right, this is happening, because you can't stop science. And I think as soon as we develop something, we will just use it. And you can't blame people for doing it, I don't think. And I don't think what I'm trying to do is try and put a stop on creativity in science or art. When it comes to a work like The Young Family, I tried to present something that was actually beautiful, but also complicated. Like, the idea that she might exist, to me, is not horrific, because like any life is valuable. And I think one of the problems with science is that perhaps we don't have the imagination to create things that aren't just bounded by our already very narrow idea of what's good and bad. And we just need a bigger space to, to create something that's even beyond that. And what happens with The Young Family is we look at her and we think, okay, she's not human, she's not pig, she's something else, but she's here obviously to serve us. And she's thinking about her offspring. We can control the technology that made her, but we can't control how she feels about her children. And if you asked me, would you cut her up to save your son? And my answer would be yes, I would do it. Absolutely. And would I put a pig heart in myself? You bet. I don't make a value judgment. But I'm interested in how that changes the way we see other animals and how we treat them, and our duty of care to them.
Yeah, I mean, I suppose it's the difficult point, isn't it? Because, I suppose the object of science, or the scientific procedure, is really about uncovering skills and techniques and revealing knowledge. And then what do you do with that knowledge? There's so many new questions that are posed by it. And when you're on a quest for scientific insight and discovery, maybe you aren't able to keep your mind, your eye, your heart on the big picture of the full implication. And of course, many scientists seem to repent almost after bringing new technologies to the world, and then seeing that actually that brings us to some great new precipice, some great new brink of potential disaster. And of course, that's not what your work is about. You're not saying this is a disaster, this is a possibility. And I suppose that's what's so interesting about your work, Patricia, is that it probably runs counter to, I would say, an apocalyptic narrative that is very deep within our culture, within Hollywood, and but tales from much further back. But maybe this is a good point, I think, to introduce Eben. And I did promise you would all understand what he did, so no pressure there. But could you introduce an anthropological view to this discussion, and maybe discuss what an ethnographies of science involves, perhaps regarding some of your research as well?
Yeah, so at its core, anthropology is about what human life is, what human life was, and also what it might be. So I'm working on a book now about CRISPR and about the stories we're telling about, speculative stories about the possible futures. So it's sort of picking up directly from what Peter was just saying about apocalypse. A word that you mentioned in response to her earlier work was dystopia. I don't really see that over in the room next door. I see a world. Patricia's created a world. It's not a dystopia. It's also clearly not a utopia. But it's a world that humans had a hand in creating, and that other organisms we're also involved in creating. As you sort of walk through the floor in there and the fleshy plant flower things start to move in response to your own movements, you get a sense that you're one agent among many that is crafting this world. So in trying to describe how we might use these technologies, I think it's critically important not to just look at ethnography of science, what is happening in the lab in a descriptive way. You know, people spend a lot, like, what is CRISPR? It's this tiny molecule you can't see. People spend a lot of their day just like moving it around in pipettes, and that's what CRISPR is, but that's not a very interesting story. You know, CRISPR is also about the informatics tools you might use, the databases that you might use to design a molecule that you want to have it do something, and often it works. More often than not, it doesn't work. So I think some of your work is about how to live with and how to care for science that has gone a little wrong, is maybe a little queer, things that come into being as a result of your actions that demand care, that might be worthy of love. So how can we live with our mistakes? How can we live with our progeny? How can we live with these, you know, we can almost think of creatures that we create in scientific laboratories as our children, as kin in some way. In some way, they're very literally kin. They share human DNA. So in addition to the humanized pigs, and I visited Shenzhen, China last year, where there's a facility that has 3,000 transgenic pigs that are more like us. A friend of mine just got a pig heart, Simon Park. He's an artist who lives in the UK. He got a xenotransplantation from one of these experiments. But how might we think about these animals differently? So during her keynote, Patricia alluded to my own sort of upbringing. I wanted to be a scientist when I grew up. At the age of 16, I worked at the National Institutes of Health, and I worked in a behavioral pharmacology lab where my daily routine involved handling rats, and these rats were being given cocaine. They were being used as experimental models for trying to understand the human condition. The big question, you know, why do kids do drugs? Why do kids do things that are self-harmful? The way the experiment was configured was really harmful. These animals had what are called catheters implanted in their head. A very painful procedure. From the catheter, there was a tube that ran down to their jugular vein, and they were put in these boxes, and you know, one of the levers produced a drug. And it's you know, those conditions of science that are critical to think about, and I think your work gives us resources for thinking differently about the relations of care that you might develop in a really confronting situation like that. But I think more importantly, Patricia's work gives us new stories to inhabit. It's not like science knows the truth and Patricia's representing it. She's giving us new stories, new aspirational mythologies that science could inhabit. This could be the science of our future. So part of my book is writing about possible futures for the human species, you know, as we use technologies like CRISPR, and there's a lot of other tools out there too. AARV is a virus you can use. Zinc fingers is another genetic editing technology that's already in clinical trials. Some of my friends are playing with these technologies, doing all sorts of things from changing racial phenotypes, things that are dangerous, that have eugenic legacies. You know, these eugenic legacies are feeding forward as people are embracing these technologies. So some of my friends are experimenting with strange things. So there's a condition that a very small proportion of the human population has called tetrachromacy or heterochromacy. It enables you to see a wide variety of colors that the rest of us can't see. So how might we inhabit new bodies? How might we inhabit new subjectivities? Are we going to care for these different kinds of subjectivities? Are emergent forms of life threatening to us? Can we live to learn with our mistakes? What might they teach us about the human condition? I think those are all really important elements of this show.
So, yeah, this is a heavy discussion, isn't it? Who knew we could get so far so fast? I don't know where to go from here. I would like to talk a little bit about Donna Haraway, the influence of Donna Haraway on your work, and obviously Patricia Donner's even written about your work, but Eben, you know her quite well, so maybe could you introduce Donna and some of her ideas, and maybe...
Do you want me to start? Or you
Oh,
start?
I'll
Whoever's
start because you know her. So I learned about Donna Haraway when I was doing film studies at Sydney University 30 years ago. And her way of understanding the world really struck a chord with me because she was talking about bodies and the way that we can see them in terms of technology. And look, that's incredibly simplistic, and that's why you're going to come in. But it struck a chord with me as a kind of young feminist. And as we've grown older, she's moved away from that. She wrote the Cyborg Manifesto, which is an important text. She's moved into looking at how we relate to other species, so moving forward together with other animals. And I've also sort of gone in that way myself. So I've looked to her. I had a chance to meet her. It was a big moment for me. But yeah, she's a very important scholar. What can you tell us about her?
So Donna Haraway wrote this rock star blockbuster essay in the 1980s that really shook up the feminist world. It's called the Cyborg Manifesto. And that essay has a punchline: I'd rather be a cyborg than a goddess. So she's basically celebrating what she calls blasphemous cyborg articulations. Not only how are these technologies of surveillance, of robotics, sort of doing the work of warfare and capitalism, but how might we appropriate those technologies and use them subversively? So more recently, she's started to write about this in sort of a multi-species or interspecies or trans-species framework, and she's written a really brilliant essay about Patricia's work. It's called, let's see if I can remember. She's got these fabulous titles. Like one of her books is like Uncomouse that meets female man at email sign. But so she wrote this fabulous essay about speculative fictions and fabulations. So fabulations are stories that we invent. So Patricia's a master fabulator. She's a master storyteller that is bringing new worlds, new futures into being. And this essay explores both the bad memory practices of science and colonialism, thinking about specifically in an Australian context, the legacies of science gone wrong. So cane toads brought here to control an agricultural pest that now disrupt ecologies and make life for endangered species very difficult. How do we live with legacies and memories of that, even while imagining new technologically mediated solutions for the future? So part of the essay really insists that there can't be science without speculation. And the stories that we tell about possible futures really guide the practice of science in the everyday. It structures grant applications. It structures the mundane hopes that one might have as a graduate student for, what do I want to work on? What can I get funding to do? So Donna is experiencing Patricia's work through the lens of love and through the lens of care. So Donna insists that these objects here are not about an erotic love necessarily, or sort of the love that one might have, that's sort of sappy and romantic, but this is an intergenerational love, a familial love that involves long-term care, lifelong responsibility. So, I think that's definitely present in your early work, and I see some really interesting themes emerging that are sort of going past what Donna was writing about. So this couple that's spooning each other right over here, we might not have the capacity to care for the forms of life that we create that might be a little bit like us, that might be a little unruly, that might be wild, that might want to, have their own worlds, have their own social spheres, have their own cocoons, have their own ecologies. So it's a beautiful essay. I believe it's for sale in the bookstore. It's a book called Multi-Species Salon. I'm the editor. It's also available on Kindle. So yeah, Donna Haraway is great.
I we really want to do some questions, but I feel like I owe you a right of reply here, because I feel like culture workers, artists, anthropologists, we've laid a lot at the feet of science that seems, not necessarily what the scientific intent is about, even if some of these problems we might see might originate from science.
I'm busting to reply.
Go for it, go for it.
So, yes, science and art, we both are inquirers of the world. In fact, Peter forgot to put this up, but we devoted
at the end, but we can
whole
we can do it
issue
now.
of Cosmos to science and art. And it's become a very popular thing. But I have to say, we are at opposite ends of the spectrum of inquiry. So I guess I want to say that. We are rule-bound as scientists. We make hypotheses based on certain rules of plausibility. We gather evidence to support or refute those hypotheses. We work on the basis that our ideas are so supportable that everybody in this room would agree if they, weigh the evidence, and if they don't, and if the hypothesis doesn't stack up, we trash it and we start again. And artists like Patricia and Eben, who I think is, quite close to that artistic impulse, are holistic. They're not bound by rules. It's the whole human experience, and it's wonderful, and we need each other. However, I feel like I want to say that I need a reality check here, because a lot of the work I've done as a science writer is dealing with the reactiveness in society as a whole to where science is going. For me, one of the most baffling things is vitamin A rice. Scientists spent 20 years to put the genes that give a carrot naturally vitamin A into rice. Why did they do that? Because 500,000 children each year in the developing world go blind because they don't have access to fresh fruit and veggies all year. But what these scientists, Ingo Petrikis, a Swedish scientist, realised is rice is pretty stable. You can have it all year round. And if we could grow rice with vitamin A in it, then parents could grow this seed, they would have it all year round, their children aren't going to go blind. 20 years it took to get into the field. What happened to that rice? Activists, Greenpeace and others bussed people into these fields in the Philippines and tore out those crops of rice. Go figure. I really don't get that. So that's what I've been trying to address, these kinds of issues very much in agriculture. And I see how incredibly powerful this anti-nature, anti-natural trope is. I mean, in this audience, who believes that natural things are best? May I see a show of hands? Yeah, it's a very powerful trope. And if Greenpeace gets up and says, GM is not natural, and it's bad for your health, even if all the major regulatory food agencies of the world have come out and said, actually, after 20 years of testing, GM crops are just as safe as any other form of agriculture, you're going to believe it pretty easily. It's a very powerful trope. So that's the real world. Good luck.
And then it gets more.
Good luck, as in getting people to accept the idea of, pig-human chimeras, and that this would be a world that people could easily accept.
I would say, we are living this world and we are accepting it. My friend Simon has a pig heart. So, I think maybe what you're doing is taking parts of our world that exist, that are sort of backstage, and putting them front stage, and again, making them a little queer, maybe perverse, but in a playful, and it's not celebratory, but it forces a confrontation with the worlds that we live, and the futures that we might inhabit.
I'm sorry, Elizabeth, that I don't need your luck, because I'm not trying to propose that that's where science should go. This is a story. It's a modern myth. It's a way to engage with a very difficult ethical dilemma in an emotional way. Because when we are confronted with real-life situations, and they're sort of very value-laden ways of talking about them, we're overwhelmed by emotion and we can't act. We can't think about these very difficult problems in a very holistic way. I don't think that's where science should go. That work, it's not about where the world is going or where I don't want the world to go. It's about discussing what might be. And I want not just scientists, but everybody here to be involved in the conversation about what science could do to make this world a better place.
What I see is curiosity, I see wonder, I see encounters involving mutual vulnerability, spaces of risk. One of my favorites is Doubting Thomas, which is right in the front room as you walk in. So you see this little child just reaching towards this orifice, and you have to get up really close to see what that ball of flesh and fur is all about, but you see these tiny little nostrils and fingers that are kind of reaching back out at the child. And how might we meet the creations of science in a different kind of way, to not just treat the animals that live in our laboratories? It's not like they don't live. This life is here, it is now, but it's discarded. At the end of the day, at the end of these experiments I was doing, the animals were sacrificed. They were put on this big heap of bodies that was in the National Institute of Health. Has this facility where it's just this big freezer, and I would go in there and there would be piles of rabbits and guinea pigs and rats, and this life and death exists around us. That's what enables our bodies right now, whether it's a physical heart in our bodies or a pharmaceutical intervention that, probably all of us here in this room have been shaped by laboratory science in one way or another. So I think you're inviting us to encounter reality as it is, not only as it might be, and be more loving, be more responsive, be more careful, be more vulnerable, open to doing the world differently, doing science differently. It doesn't have to happen that all these animals get killed. There's a remarkable group that's sprung up in New York City. A lot of people in New York don't like rats, and you see them scurrying on the subway and might even brush your leg as you're waiting on the platform. But there's this group of 512 odd people who rescue rodents, and they rescue lab rats and care for them. So what new practices of care and responsibility might we develop in response to Patricia's art? I think that's something that not only scientists should take away from this show, but that all of us can take away. How might we live our own lives differently?
These are some emotional discussions, aren't they? And I mean, look, I want to add as well, of course, science isn't the only venue in which animals are mistreated. There's agriculture, there's even how we treat wild animals, even questionable domestication practices, and of course our relationship with animals is not just in the present. It reaches back centuries, eons. So there's big discussions. So please, this has been a great conversation. Patricia, Eben, and Ella, please help me thank them.